So, picking up with part two of the muscles that we were working on the other day, I think we left off with myasthenia gravis as the last portion of what I had spoken about. So, moving on. So, before we get started again, remember that there are some important players basically in the fibers that are going to be part of the muscle contraction. So, you have the thick filament down here in the purple, which is called myosin. The myosin has these cross bridges or myosin heads that are going to interact with this light purple, whatever lavender color that is. That is the thin filament, and the thin filament is made up of the little spheres, which are the actin, the fibers, which are the tropomyosin, and then those little bitty proteins that are actually the troponin. So we have to have a couple things occur for an actual muscle contraction to occur. The myosin, the thick filament, is going to have to come in contact with the actin when your muscles are relaxed. That tropomyosin, that long thread, is preventing that action from occurring. So as we get started, these guys are going to have to be, sorry, the cat's going nuts. These things are going to have to start working together. All right. So, reminder, SR is the muscle's equivalent of the ER, and this is where you store calcium. So remember at first we had calcium that at the end of, here's my little nerve cell, when the nerve impulse came down, calcium channels open, calcium flows into the nerve cell, and it causes the release of the neurotransmitters, right? That's the first role of calcium in a contraction. The second role is going to occur now, and it occurs at sort of this level down in here, okay? So calcium, let's go back, sorry. Here's my little nerve cell. Here's my muscle cell surrounding it. We have transmitted chemically, we have moved this impulse from electrical impulse to a chemical impulse, and now the nerve cells on the other side, the muscle cells on the other side need to generate some activity. So as the little channels open up, sodium flows in, we're going to have a generation of an impulse. That sodium and potassium and stuff that's interacting right in here, that little impulse that's in that muscle fiber now is eventually going to reach that SR. So if I have a muscle here, remember I have those little triads here, and then in between I have this little network of the SR, okay? So the calcium is stored in there. And once the information gets to this point, calcium that is stored in the triads, in that little SR, is going to be released out of that and actually come into contact with those fibers there, okay? So it's leaving its storage place, and now it's coming in contact with those fibers. And what it does is it binds to those little bitty proteins, the little troponin proteins right there. So here's my little troponin proteins. When the calcium binds to the troponin, it causes what we call a conformational shape change. So let's start right here and look at that shape. Notice the tropomyosin is on the outside. It's blocking that actin. It's keeping the actin from interacting with the myosin heads right here. When calcium binds to the troponin, that moves those fibers towards the inside. Notice they're on the inside now, and the little spheres are faced outwards. They're on the outside. They can now come in contact with the actin. So my little actin comes in contact with my myosin, and we're going to have what's called a stroke, like an oar stroke if you were rowing or paddling or something. These guys bind to the actin, and then they just pull backwards. So here it is binding. Then you have right here binding, action going across, and notice they bend. And as they bend, they're really moving this fiber that direction. So this binds, moves that way. They release, they pick up another one, and then they move that fiber again. So you just have this sort of This a little bit of action of moving the actin, moving the actin, moving the actin. The myosin never shifts positions. Okay. So binding sites exposed. And that's this point right here. So binding sites are exposed. The actin and myosin now come in contact with each other. That forms those little cross bridges, an actual physical bind together of the two. So here's the actin. The myosin comes in and binds onto it, and it will pull on this. So if we look at the actin here, myosin head attaches and pulls, and then it releases, attaches, and pulls. So it's always pulling that actin in. Okay. Okay. So here again is another view of this. Remember definition again, sarcomere from Z line to Z line, that is one contracting unit, and one contracting unit is called a sarcomere. When the actin and myosin interact, so the center section here that I'm drawing in right now, that's myosin. These thin fibers, that's the actin. When they interact and those little myosin heads come in contact with the actin, they force this to move this way. And then these on this side, we're doing the opposite, and that's forcing that to move in. So notice the space here gets smaller because the actin's moving in. The width of the myosin never changes. Here's my myosin, same width. That stayed the same. The only thing that changes during this contraction is the space between the end of that actin and that actin there. It got smaller. So this whole thing from here to here moved inwards together, all right? So the only things that we see, Z line to Z line, that moves in. That's a muscle contraction, okay? I band, here's my I band here to the other side here. That also changes. Notice we had a longer width here. Now we have a shorter distance here. So that gets narrow, okay? And then the H zone. The H zone is right here to here. So this is the H zone. The H zone is also going to get smaller. The length of the fibers never change. It's spaces that change. I band gets smaller. H zone gets smaller, and that's about it. And then you have a fully contracted muscle here, okay? So as long as calcium is moving in, calcium is binding to that troponin on the thin filaments, then you're going to have this movement, these stroking actions basically of a paddle, moving these two fibers closer together, okay? So as long as that calcium is present. If I want to stop that action, I now need my muscle to relax and go to its original position, which is right here. Then you're going to have to remove the calcium back out so that that troponin comes off of the actin, and you can go back to your original position with the troponin and the tropomyosin on the outside. Remember that anything like this, those are links to things, and I put this one up here, sliding filament model. I put that link in D2L for you. All right, so more cross-bridge cycling. Remember, this is the sliding filament theory that we're describing. Don't forget that term. Sliding filament theory. Okay, all right. So myosin cross bridges. That's the myosin head, cross bridge when it's attached. That's happening right in here, cross bridge. Attached to the actin. When it does, it pulls, forces that actin to pull the actin, or forces the myosin, forces the actin pull that direction there, okay? It releases, and then it will attach again. All of this is requiring energy. So you know that ATP is going to be used here. So ATP actually binds to a myosin head, and then it stores the energy in that myosin head, and the ADP then is going to be, what after you take the energy from ATP, you release a phosphate and it becomes ADP. So that ADP and the phosphate is then released from that myosin, and the energy is stored in that myosin head to create that movement, to allow that movement to occur, to give the energy for that movement to occur. All right. Then we have to have a new ATP molecule. Once that energy has been used, you got to have a new one. So new ATP binds to the myosin. That breaks your linkage between the actin and myosin. So what we had here with this linkage and this stroking motion, it breaks it, and it's going to return it back to kind of this position up in here so that it can reattach again. ATP splits, store the energy back in the myosin head, and start back over and create another stroking action and move that actin in a little bit more. And again, animation, this link is in D2L for you. I would definitely look at this link. Everything I just described and tried to draw for you, that's actually being shown in animation form right here. So you can see in green, they've got this is the myosin. Here's the myosin heads, and it shows how the ADP and ATP are used. You've got the little actin spheres and the tropomyosin threads, and then this is the troponin right here. So this is all being described and shown to you. So I would definitely take a look at that video. It's going to do a lot better than my drawings did. All right, so I've contracted my muscle. Now it's time to release and relax, because you can't keep your muscle always in a contraction. At some point, you're going to have to relax that muscle. So here's the thing. If you remember acetylcholine back here, and let me draw my little, there's my nerve cell, right? The nerve impulse came in, calcium channels open, calcium flows in, and my neurotransmitters, that's ACH, move to the surface of that nerve cell, and then they cross that, it crosses that gap. Okay, y'all with me on that one? As long as ACH is bound to the receptors, these little protein channels on the other side, as long as I have them bound, this actual, let me try that again. So here, and then here's my little channel, and it's open. As long as my neurotransmitter is bound here, this channel will be open and sodium flows in. As long as sodium is flowing in, it's going to lead to that cascade, and you're going to see a muscle contraction. So if I want to stop that muscle contraction, I need to do a couple things. I need to stop the nerve impulse. You need to stop the flow of calcium in, acetylcholine leaving the nerve, the end of the nerve cell, and binding to the muscle cell. So that's one of my points of control right here. As long as acetylcholine is bound, remember, to this protein channel, this is going to be open, and it's going to be telling that muscle cell to contract. So what do I have to do? I have to close this channel, and the only way to do that is to rid myself of the acetylcholine that's bound here. That's going to be an enzyme that does that. So acetylcholinesterase, really long word, guys. Here you go. ACH, ACE for short, acetylcholinesterase. Takes that acetylcholine, removes it off of there, shuts this channel back, and now my contraction is going to quit. Okay, so you really need, not only you need acetylcholine to make a contraction occur, you also need acetylcholinesterase to stop the contraction. So this stops the contraction so that you can have relaxation occurring. Okay, that's one of the points of control, and it's a very important point of control. Think about what would happen if the acetylcholinesterase, if you destroyed this molecule right here, you would forever have a contraction. That's called tetanus, when everything contracts up and you can never relax. This is also something that occurs in different venoms. So some of the venoms that attack us, some of them are called hemotoxins and some are called neurotoxins. Hemotoxins actually go in and like destroy the cells. A brown recluse is a hemotoxin. A neurotoxin is what a lot of the vipers are, and black widows. So they create a neurotoxin. And basically what they're doing is they're stopping, they're destroying this acetylcholinesterase. If they destroy that, then you can never stop a contraction. So basically you just continue to contract. And that includes, it's not only painful with the muscles, but your diaphragm continues in contraction, so you can't breathe. So it causes death through the lack of being able to breathe. And that's how those venoms work on that. Other venoms work in other ways, but that's one method. We also have some pesticides that work that we're used to. We took them off the market. Pesticides that work that way. So you would spray your plants with the pesticide. The insect that uses the exact same whole model, this whole, the whole acetylcholine, everything model for their muscle contraction. They would ingest the pesticide. It had acetylcholinesterase destruction factors basically in it, so it killed their acetylcholinesterase. So the insects would twitch to death. Unfortunately, people don't read instructions, and the big warnings on the outside of those pesticides said wear gloves, wash your hands. People didn't do that, so it had the same effect on individuals. It was a kind of a dangerous situation to be in, so they had to take them off the market. But it was very effective in killing insects. It's just that, unfortunately, we work the same method that they do. But okay, back on track. So if we can stop this action, so close that channel right there, that means removing the acetylcholinesterase, removing the acetylcholine with the acetylcholinesterase, and it basically just returns it back up into that neuron up there. Then you will shut this down. You're going to remove the calcium. Now the calcium isn't getting that information from here, from the sodium influx that says, hey, go in and attach to the troponin. So that moves back to the SR right here. Okay. If the calcium's back in the SR, then the myosin and actin can no longer bind together, and you're going to have that relaxation. So there's, like I said, there's a lot of points of control. Think about that. I told you about if there was no acetylcholinesterase. What happens if there was no acetylcholine or not enough? Then even though you got a nerve impulse coming down, you got calcium flowing in, you're not going to have the next step, which is the acetylcholine moving across to tell the muscle fiber to do something. So it's like a dead end. You get all this interaction and this message coming in, but you get no muscle contraction. What happens if your calcium levels were low? Well, you've got 2 points of control for calcium. Calcium is the thing that has to tell the acetylcholine to leave the neuron, but calcium is also the substance that binds to the troponin that then allows the actin and myosin to interact. So if my calcium levels are too low, then there's no binding there, and you will never get a contraction. So, think about all of those. Everything has to work just right. And you have to have, this is why it's important to have the right amount of things in your body. And in calcium, we think of calcium as being this important thing with bones, but it does strengthen bones, but it's also really, really important with muscle contraction. Without it, you're not going to have muscle contraction. So it's one of those really important substances that you need in your body, and that's why you need to maintain proper calcium. Your body will still the calcium. If you have calcium stored in your bones and you're not getting enough to go through this action of muscle contraction, think diaphragm, not just your movement, but diaphragm and heart and all of that need this too. If you're not getting that calcium for that, you're going to steal from your bones, and that'll weaken your bones in the long run. So always maintain enough calcium in your diet. All right, so talked about energy. Energy is in the form of ATP. So we have these ways of having it available to your muscles at all times, because without the ATP, you're not going to have a lot, or you're not going to have enough energy to allow for those contractions to occur. So if I have a lot of ATP available, we've been making a lot of ATP is high, then really I don't really need it. And remember, ATP doesn't store, so I'm kind of wasting it. So what we want to do is we kind of want to make that energy storable for later use. And what you're going to do then is you're going to store it in the form of this creatine phosphate. Okay, there's that little phosphorus, and you can always transfer. Notice it transfers back and forth, the phosphate back to an ADP and make ATP when you need more ATP, or you can store it away as creatine phosphate. So we have these two energy sources. Not only do you need to think about ATP, but creatine phosphate, and the important part is the phosphate right there, stores energy that you can then turn into ATP very, very rapidly as you need it. And then you can always make more ATP through cellular respiration. But that takes a little bit of time, to go through step 2. You got to go through all that glucose and glycolysis and Krebs cycle and all of that. If you've stored your phosphate onto this, made this molecule of creatine phosphate, you can quickly access it and add it to that ADP and then make an ATP real quick. So that's one way to get access to ATP. You still have to go through cellular respiration though. All right, so reminder, all that cellular respiration we went through earlier in the semester, here it is again. You have anaerobic respiration, which is that glycolysis, breaking down the glucose into smaller and smaller units, and we eventually get carbon dioxide out of it that you release, but what you really get is ATP through the whole process. So the anaerobic portion is that glycolysis in the cytoplasm. A little bit of ATP. Not much. It's not going to get you very far. You really need to continue from, here's glycolysis right here. You really need to continue into Krebs cycle and then the ETC to get a whole bunch of ATP out in the end. That part is what we call aerobic. Krebs cycle and ETC that's happening inside the mitochondria. That is, that's the aerobic portion, and that's where you get the majority of your ATP. So this is where we really want to get to. That's going to be the best way of getting enough muscle energy available for those contractions. So remember I showed you at the beginning that a lot of the muscle fibers have a whole bunch of mitochondria around that, and that's because of this need for this ATP. We also have this little protein called myoglobin. Myoglobin gives your muscles this sort of darker red color, and it stores oxygen. So it makes oxygen available because oxygen is going to be used right here at this final step in the electron transport chain. And that's, again, where we get the majority of our ATP. So it's really important to have oxygen that you're breathing, but you want to have it sort of on hand. So you breathe it in, you store it in the muscles in this substance called myoglobin, and then that oxygen can be released out and used for cellular respiration as needed. Okay, sometimes you go from, probably doing that a lot these days, sitting on the couch, being inactive, and then suddenly you require energy. You're walking, you're running, you're moving about, okay? So we use that oxygen that's been stored in that myoglobin. So we have that oxygen that was stored in the myoglobin. It gets used up pretty quickly, and I'm going to need more oxygen. Well, the simple thing is to start breathing again, but you still have to go from your mouth to your lungs to your blood vessels to your muscles. So it takes a little bit of time. So we use up the available oxygen that you had stored away, O2, in the myoglobin. And I haven't quite got enough oxygen there from breathing. So we enter into what's called oxygen debt. So we're using more oxygen than we are actually able to provide to those cells. Yet those cells need a lot to produce ATP. So oxygen is not available. We're going to continue with glycolysis because remember, glycolysis is anaerobic, so it doesn't require oxygen at that point, and it does give you a little bit of ATP, not a lot, but some. But instead of going from the pyruvic acid to Krebs cycle to continue with its processing, because it can't do that without oxygen, it's going to go through that lactic acid fermentation, and we went through all this in chapter 4. But you've got the lactic acid fermentation, but this allows your body to produce, produce some amount of ATP to keep the cells alive for the time being, okay? But you're in oxygen debt because you really don't have enough oxygen to keep this going on forever. But once you get enough oxygen, start breathing really heavy, get a lot of that oxygen, the oxygen finally makes it to those cells, then I can take that lactic acid that I produce that caused your muscles to burn, send it back into the glucose form, so back up into here, and then go through that whole process and enter Krebs, enter ETC, and really get a lot of oxygen. But until you can do that, you're in oxygen debt, really, to your body. Okay, once you've been working out a lot, you're going to end up possibly, depends on what good a shape you are in. The more you work your muscles, the better off you are. But we can have different types of muscle fatigue. That's just basically tiredness. So if I'm tired, I'm not going to be able to contract that muscle very well. Can be many different things. Decreased blood flow. Remember, blood flow is going to give you oxygen. It's going to give glucose. If I don't have oxygen, I don't have glucose, I'm going to reduce my amount. And coming from the blood, then you're not going to have the amount of ATP that you need. So that's going to affect your muscle contraction because it's not getting what it needs to do that. Ion imbalances are also a problem. Ion imbalances are really important because remember, I talked about calcium, talked about sodium, didn't really mention it, but there's another one called potassium. All of these ions are really important in both nerves, so the nerves and in conducting the impulses, and in your muscle contractions. So if any of those are too low or even too high, you're going to have problems with contracting those muscles. So again, it's very important to make sure that you have the correct amount, not too much, not too little of those ions in there. And then lactic acid from lactic acid fermentation is also going to affect your muscles. This is when you first start exercising and you get that really burn sensation. It's not the day after soreness. This is that burn that you get and you think, I just want to quit right now. But if you keep running or keep moving, it kind of goes away. But that lactic acid does cause the muscles to be tired for a short term. Okay. Then we have more sustained things like a cramp. Cramp, this guy up here in this picture has got that. This is a muscle contraction that just doesn't stop contracting. And it's not to the worst part. The worst part would be what we call tetany. It won't stop and you can't get it to stop. Cramp is a sustained contraction that doesn't stop, but eventually, you rub it, you do something, you move your muscles in a different way, you can get that to relieve itself at some point. Tetany, you really can't. It's impossible to really stop it without medical attention most of the time. All right, and then there's physiological fatigue. That's just your muscles aren't used to doing the work that they're doing. They don't have enough mitochondria to provide enough ATP. So you just really can't overwork them more than they can do that. But with regular exercise, you do a lot of things to the muscles. The muscles will respond to exercise. They build more ATP factories. They build more mitochondria. Say, I need my muscles to work more. I've started an exercise routine. Your muscles respond by saying, hey, we need more energy. We need more mitochondria. We're also going to need more blood flow because we need oxygen and glucose. So you build more capillaries to those muscles. And you can train yourself basically you train those muscles as they get more and more exercise to be less likely to go through lactic acid fermentation and produce a lot less lactic acid. The really trained athletes really don't produce a lot of lactic acid when they're working out. Their muscles are much more likely to hit up the mitochondria for the energy that they need. And they have a lot of mitochondria because their muscles have been used to having to have that. Muscle contraction is one of the main ways we get heat production in our body. Remember, through that whole cellular respiration process, one of the byproducts is heat. So every time you start to make ATP, you're going to make heat. And your muscles produce a lot of ATP for muscle contraction. That means you're producing a lot of heat. Most of that heat is going towards just your generalized body temperature, 98.6-ish, right? Not really, but kind of in that range. A little bit above, a little bit below, but that's the sort of range in there. That's that most of that body heat, that body temperature is coming from muscle contraction. Anything excess, you're going to sweat it out. You're going to release that heat out, but it is the main source of maintaining our body temperature. And then wherever it's made. So I'm working out my muscles and my legs, but I'm not doing my upper body or something. Your blood is still going to transport that heat and just distribute it throughout the body so that, it's all over in different areas. All right. We can look at and we can measure, scientists measure muscle contractions. So we can look at a muscle contraction and we can look at a single muscle fiber and we can kind of measure the force, the strength of one individual fiber. Now, one fiber by itself isn't going to have a lot of force or strength, but you put a whole bunch together in a huge rope, that would be your muscle as an organ, and we do get a lot of strength. But we can take it out. We can look at one little fiber and say, how much does one little fiber contribute to the whole thing? So you have these little devices, and basically you just hook it up, like one end, and just kind of stretch them, and then look at the changes in the overall length of the muscle and see where it is that basically what is the optimum, I guess is what I'm looking for, is the word there. All right. So here's what we need to look at. What is the minimal strength required to cause a contraction? This is something we call threshold. All right. Threshold is this. I can send information like, you can release the calcium, you can release that sodium, you can do all of those things. But until you reach a certain point of that, you may still not have a contraction occur. Once I reach a desired level, that contraction is going to occur no matter what I do. We call that threshold. So I can do a little bit of work here, but until I reach this point, nothing occurs. Once I reach that point, I can't stop it. Okay? Minimal amount that is required for a contraction to occur. This is what the definition of threshold is. You're also going to see threshold when we talk about nerves specifically. All right. A twitch. Here's another term for you. A twitch is an actual little contraction. So every time your muscle contracts, that's actually a twitch. Okay. So as we look at it, and you can record it, here it is being measured, right? So here's my measurement, and you can look at the bottom. There's time of the stimulation. Here's my force. That's what we're measuring in this twitch. So as I'm moving along right here, you're relaxed. Nothing's occurring. At this point right here, it gets some sort of information from a nerve that says, I want you to contract. But it takes a little bit of time. See that little time from here to here? It takes a little time to actually react to that. This is our latent period. So it's getting the message. It just hasn't been able to respond to it yet. Okay? So the latent period. Once it starts responding, then you're going to start seeing muscle contraction. So this is the force going uphill from the muscle contraction. That's our period of contraction. All right? Reaches a certain point, enough of the contraction, it's time to relax. And so we're going to go back down here and less and less and less and less force until we get back down to normal. So this peak is just increased force and then decreasing that force as we go down when it relaxes. All right? So period of contraction to the peak, period of relaxation from the peak back down to its original relaxed state. I'll need to send at some point, another contraction again, but I can't do it right away, okay? There has to be a time period. You can't just keep like on top of each other immediately. It has to come down a little bit before it can even go back up again. We call that refractory period. So it's a period of time when the threshold stimulus, it's not going to send it. It doesn't matter if you send information and your brain says go ahead and twitch. It's not going to respond, okay? No matter what information it's getting from the neuron. It has to spend a little bit of time sort of recovering before another twitch can occur. Another term in here I want y'all to be familiar with is all or none response. Our muscles either contract or they don't contract. They get a message or they don't get a message. They really don't work harder. I can't tell that twitch to go more and do more work. The only way I can get more work is if I recruit more fibers. So if I want more force, I get a whole bunch of fibers. If I want less force, I just tell a few fibers to do it. But each fiber is going to have the same force. It's going to have the same amount of information that's being sent. Does that make sense? It either does it or it doesn't do it, all or none. It doesn't do it more or less just because I want it to. One of the things I use, and I'll talk about it, probably I'll bring this up when we talk about nerves too. When you flush a toilet, you can start pushing on that handle of the toilet and you can hear the water going, but it doesn't flush yet. Once it reaches that point where it flushes, that's what we call threshold. So it reaches that point and it's flushing. You can't stop it at that point. You have hit that point and the toilet's going to flush. Threshold has been reached. You can't press on that handle harder or less to get it flushed to a little bit less or a little bit more. It either does it or it doesn't do it. Once you reach threshold, it's going to flush. If I need more force behind it, I'm going to have to flush it again later, right? And there's that refractory period. You can't flush right away. You've got to wait till it refills and goes through its little period again. So that kind of gives you that point of time for that. All right. So sort of something to, I guess, link that thought to. All right, length tension. When we, let me just stop this.